All right, so this is all about transduction. And first thing we want to talk about is I can define transduction and the concept of a cell signaling pathway. So to transduce means to change forms. And so when we talk about transduction, what we're doing is we're changing the signal from the form of the ligand, which is reception, to the form of the response. So everything between reception and response is called transduction. And it's just changing that form. This is all old stuff. You don't need to pay attention to that. But it's just changing that form from this type of signal to this. So from, an adrenaline molecule to a gene that's being expressed or whatever. All right. It says, I can explain the process of amplification within a cell signaling pathway. Okay, what happens in a cell signaling pathway or in transduction is that there are generally more than one step. So a lot of times you'll have, 3 steps, 12 steps, 100 steps. It just really depends on the pathway. Every pathway is a little bit different. But what can happen with amplification, which just means to make bigger, is we have this molecule number 1 and it's activated from some previous step, reception or whatever. Let's say it's a kinase, and so it can then phosphorylate. So we'll add a phosphate group to number 2, which activates it, okay? As soon as that one's activated, it breaks off to do its next thing, and number 1 is still working. So number 1 can then phosphorylate another number 2 and activate it. Okay, that one breaks off and does its job, and so it phosphorylates another number 2 and activates it. So now we've gone from having one pathway to 3. Now let's say that each of these number 2s, let's say this one can phosphorylate, let's say it's a kinase, and it phosphorylates number 3. Okay, and then so that 3 goes off and does its job, and then it can phosphorylate another number 3 and activate it, so that can go off and do its job, and then it can phosphorylate another number 3 where it can activate it and go off and do its job. Okay, and let's say that each of these 2s, activates 3 3s. Okay, and so now what we have, instead of having one pathway, we had 3. Now instead of 3 pathways, we have 9. And then imagine if all of these 3s were to activate 3 more, we'd have 27. And so you can see as you have more and more steps, and each of these proteins stays active long enough to do its job several times, you can see that it amplifies really quickly. And so you can get a really big response. Instead of just one pathway giving you one response, you can get 27 or 81 or whatever, however many responses. So that's the process of amplification within a signaling pathway. I can describe the function of a kinase. A kinase is simply something that adds a phosphate group, which is going to activate. So for most molecules, when you add a phosphate group, there's always exceptions, but for most, when you add a phosphate group, that is going to activate. It's like an on switch. It flips that molecule on. So a kinase is going to add, and so it activates, or we can say it causes activation. So it says, I can describe the function of a kinase. It adds a phosphate. That is called phosphorylation. We've talked about this before in Unit 3 when we talked about substrate level phosphorylation and oxidative phosphorylation and photophosphorylation. It is simply the addition of a phosphate group. Normally in those scenarios, we were adding a phosphate to ADP to make ATP. Here we're adding a phosphate group to any kind of thing. It's not necessarily ADP. A lot of times we're adding a phosphate group to another kinase to activate it so that it can add a phosphate group to something else. So I can describe the function of a kinase. It adds a phosphate group. I can explain the results of phosphorylation. It activates the molecule. And then the opposite of a kinase is a phosphatase. And I always want to spell this wrong, phosphatase. It is going to remove a phosphate group, which is going to be, that's called dephosphorylation. And that is going to deactivate. It's going to turn that molecule off. So let's say that we have an adrenaline rush. We get scared, we think that something's coming to get us, and so we want to phosphorylate, we want to go through transduction, we want our adrenaline to get a response, which is going to make us move faster, breathe faster, our pupils will dilate so we can see better, all those things. But then we realize, oh my gosh, that was just a cat making noises, in the backyard, and it scared me. I thought somebody was trying to break in my house or whatever. And so I realized, it's no big deal, it's just a cat. Well, I don't want to keep having that reaction. I need that to stop. And so what you can do is you can deactivate or you can terminate that pathway. And one of the ways we do that is by going back to all those molecules we added phosphate groups and taking those phosphates off. That's going to turn off all those molecules. So imagine if these were all phosphorylated, it's going to turn all of these off. So all of these pathways just stop. The next thing is I can explain the role of second messengers, and I can give examples of second messengers. This is going to be easier for me to talk about with the PowerPoint. And so a second messenger is just anything after the first messenger, which is the ligand. They are small and they are not proteins. They are polar. And the reason they're small and not proteins, that's not important as much as the reason they're small and polar is because they are in the cytoplasm. The cytoplasm is a water-based solution and water is polar. So anything that's in the cytoplasm is going to need to be able to hang out or handle being in a polar scenario at least sometime. What a second messenger is going to do, because it's small, and this doesn't do a great job of showing you how small it is I mean, it does show it's smaller than this guy, but it's generally a good bit smaller, is because it's small and it's polar, it can like zoom. Good afternoon. Sorry, we got interrupted there. But a second messenger is just tiny and it's polar, so it can just zoom through the cytoplasm a lot faster than this big guy. So it's just going to speed up the reaction. And the two examples that you need to know are CAMP, cyclic AMP. So that's like AMP, ADP, ATP. That's the AMP. And it's cyclic because it can cycle back and forth between ADP and ATP and AMP. But that's one, and then calcium ions are another. There are others, but those are the two you're most likely going to see. And then the last thing is I can talk about the benefits of a multi-step pathway, and I think that's in here somewhere too. Maybe not, but multi-step pathways, again, to allow us to amplify. We've already talked about that, but they also allow us to regulate it. So for example, if I go back to my multi-step pathway over here, if I decide, oh, I don't need this anymore, I can shut it down at step 2. And that doesn't even give me a step 3. Or if I'm already past step 2 and step 3, I can shut it down at step 3. So you can imagine if there's like 10 steps, there's 10 different places I can stop it before I get to the end. And so that is going to help me regulate it a little bit better, a little bit more specifically. And so that is how I can, or those are the benefits of a multi-step pathway: amplification and easier regulation. And I think that's it. If you have any questions, let me know.